Hello all. Welcome back to another video tutorial. Today's topic is renal clearance of nanoparticles. As we all know that, nanotechnology has incredible potential to help us detect and treat diseases in our bodies. But there's a challenge. Many of these tiny particles have an electric charge that makes it hard for our bodies to get rid of them easily. If they stick around too long without being cleared out or broken down into harmless parts, they could become harmful to us. Imagine these nanoparticles as tiny messengers carrying important packages of medicine or information. But if they hang around in our bodies for too long, they could start causing trouble. So, scientists are working hard to figure out how to make these nanoparticles easier for our bodies to deal with. They want to find ways to help our bodies clear them out or break them down into safe pieces. That way, we can enjoy the benefits of nanotechnology without worrying about any potential harm it might cause. Understanding how nanoparticles interact with healthy tissues and organs has led to new ways to control where they go in the body, which is just as crucial as targeting specific diseases. In the liver sinusoid, nanoparticles slow down significantly, reducing their velocity by 1,000 times. This slowdown means they stay in the body longer, which can be both beneficial and challenging. Understanding how nanoparticles interact with biological systems helps improve how we target diseases and how effective treatments are. It also helps ensure that nanomedicines are cleared from the body efficiently, which is crucial for their safety and effectiveness. Kidneys are the second major organ for nanoparticle clearance. They filter nanoparticles from the blood and quickly remove them from the body. By adjusting the size, shape, and surface chemistry of nanoparticles, scientists can control how they interact with different parts of the kidney. Understanding these interactions helps improve how we target diseases with nanoparticles, manage their movement through the body, and reduce any risks they might pose to our health. To learn about how nanoparticles interact with the kidneys, it's important to understand the anatomy and function of the kidneys. Let's start by exploring the basic structure of the kidneys and how they work. The kidneys are positioned in the retroperitoneal space along the back wall of the abdomen, one on each side of the spine. Typically, the right kidney sits a bit lower than the left due to the presence of the liver. Shaped like beans, they receive blood from the renal arteries and send it back out through the renal veins. Urine produced by the kidneys travels to the bladder through the ureters. The kidney has three main regions: the renal cortex, medulla, and pelvis. The nephrons, found in both the cortex and medulla, are the kidney's basic units. Each nephron consists of several parts, including the renal corpuscle, proximal tubule, loop of Henle, distal tubule, collecting ducts, and peritubular capillaries. There are two types of nephrons: cortical nephrons, located in the outer cortex with shorter loops of Henle, and juxtamedullary nephrons, situated near the corticomedullary border with longer loops of Henle. The afferent arteriole brings blood into the renal corpuscle, specifically to the glomeruli, where high blood pressure forces fluid, solutes, and waste to be filtered into Bowman's space. Any unfiltered fluid then exits through the narrower efferent arteriole, entering the peritubular capillaries and eventually the renal vein, rejoining the bloodstream. The waste that was filtered flows from Bowman's space into the proximal tubules. These tubules have a surface covered with densely packed microvilli, facilitating the balance between reabsorption and secretion of urine. Kidney filtration is facilitated by the glomerular filtration membrane, which is supported by mesangial cells within the mesangial matrix. Mesangial cells, situated between the glomerular capillaries, also play a role in regulating the filtration of nanoparticles. The glomerular filtration membrane consists of several components, including the endothelial glycocalyx, endothelial cells, the glomerular basement membrane, and podocytes. The endothelial glycocalyx, composed of glycosaminoglycans and proteoglycans, helps prevent protein leakage and contributes to kidney filtration. The endothelial layer contains fenestrations openings of 70 to 90 nanometers, along with the glomerular basement membrane, primarily made of type 4 collagen, laminin, and proteoglycans. This network forms pores ranging from 2 to 8 nanometers in size. Podocytes, located on the other side of the glomerular basement membrane facing Bowman's space, have pores sized between 4 to 11 nanometers and are covered by a glycocalyx approximately 200 nanometers thick. The unique four-layer architecture of the glomerular filtration membrane means that kidney filtration depends not only on particle size but also on charge. Nanoparticle interactions with glomerular barriers. The efficiency of renal clearance for macromolecules and engineered nanoparticles is influenced by various factors, including their size, charge, shape, composition, and interactions with different kidney compartments. Size dependency. Nanobio interactions and the glomerular filtration of nanosized macromolecules are significantly dependent on size due to the distinctive ultrastructure of the glomerulus at the nanoscale. Various engineered nanoparticles have been developed, but their efficient clearance from the body relies on understanding how they pass through the kidney's filtration system. The kidney filtration threshold for metal-based nanoparticles is around 6 nanometers, determined through studies with quantum dots. This means that particles larger than 6 nanometers tend to accumulate in organs like the liver rather than being cleared by the kidneys. Even though larger nanoparticles usually accumulate less in the kidneys, they can still interact with the glomerulus. Many studies have shown that non-renal clearable gold nanoparticles coated with PEG and ranging from 43 to 167 nanometers barely pass through the endothelium if their size is over 100 nanometers. However, particles smaller than 100 nanometers can pass through the endothelial layer but are blocked by the glomerular basement membrane, leading to their accumulation in the mesangium. It's worth noting that some large nanoparticles can break down into smaller fragments either in the bloodstream or within the glomerulus, eventually being cleared by the kidneys. Nanoparticles with a hydrodynamic diameter larger than 100 nanometers are unable to pass through the endothelium. Those with a hydrodynamic diameter between 6 and 100 nanometers cannot cross the glomerular basement membrane. Particles with a hydrodynamic diameter of 2 to 6 nm can traverse the glomerular filtration membrane, with smaller particles crossing more rapidly than larger ones. Nanoparticles with a hydrodynamic diameter of 1 to 2 nm pass through the membrane at a similar rate. Those with a hydrodynamic diameter smaller than 1 nanometer physically interact with the endothelial glycocalyx, resulting in inverse size-dependent filtration, where smaller particles are cleared more slowly than larger ones. Charge dependency. Surface charge plays a significant role in nanobio interactions and the transport of engineered nanoparticles in the kidneys. Upon entering circulation, nanoparticles often have proteins adsorbed onto their surfaces, altering their charge and size. In terms of glomerular filtration, positively charged gold dendrimer composites are cleared faster by the kidneys than negatively charged ones of the same size. Similarly, cationic quantum dots pass through the glomerulus more quickly than anionic ones. Anionic quantum dots can traverse the glomerular endothelium but accumulate in the kidney due to internalization by mesangial cells, likely influenced by charge-dependent repulsion at the glomerular basement membrane. Conversely, the electrostatic attraction between the glomerular basement membrane and cationic nanoparticles may accelerate the renal clearance of cationic particles, even those typically not cleared by the kidneys. Positively charged nanoparticles exhibit faster clearance compared to neutral ones, followed by negatively charged nanoparticles. This trend is primarily due to interactions with the glomerular basement membrane. Additionally, highly negatively charged nanoparticles are cleared more rapidly than nanoparticles with lower negative charges. Other factors which influence the renal clearance are shape and composition of the nanoparticle. Thank you for watching, and stay tuned for more interesting topics.